Hello everyone. Today we will look at the introduction for metabolism. A quick definition from Wikipedia for metabolism is the set of chemical reactions required to sustain life in an organism. Metabolism can be divided into two main groups, anabolism and catabolism. Anabolism is the synthesis of metabolites and is known as biosynthesis. Catabolism is the degradation of substances, and the process is known as oxidation. The currency to conduct metabolism in the cell is ATP. This is the structure of ATP. I'm sure you all know the structure. Do remember that GTP and other nucleosides are equivalent to ATP. Metabolism is a set of pathways, which are reactions that occur in a certain pattern. There are three types of pathways. An example of linear pathway is glycolysis. For cyclic pathway, a good example would be citric acid cycle. And the last type of pathway is branch pathway. An example of this is purine synthesis. Some of you might not be aware of this, but we'll learn it very soon. In this diagram, the capital letter A would be called the substrate, and the capital letter E would be called the final product. Capital letters B, C, and D are known as the intermediates, whereas the B, C, D, and E in small caps are known as the enzymes which catalyze the specific reactions. Compartmentalization is a big theme in metabolism. You need to have separate anabolic and catabolic pathways, and you want one of the other. You do not want to break down the same material you just formed. Hence, you need to have distinct enzymes which control these reactions. Some enzymes can actually conduct the reversible reactions, which we'll explore in more detail at a later time. Some key advantages of compartmentalization is that it increases the effectiveness of your pathways. But it does require specific transport proteins to move your substrates and metabolites. The distance required for these metabolites is reduced if a process is in one particular area. How do you study metabolism? In an experiment, you can try to measure the concentration of certain metabolites, or you can look at the physiological effects on the organism. How relevant is your in vitro experiment to what happens in real life? Where do you conduct your experiment? In an entire organism or just the mitochondria? These are some of the questions you would like to keep in mind when you learn these processes. Thank you. If you have any questions, please visit the Contact Us page and enjoy the course.